Hi, I am going to present the article Cycloartobiloxanthone induces human lung cancer cell apoptosis via mitochondria-dependent apoptotic pathway. Before starting, I want to mention that I live with my friends, so sorry for the background noise if there are any. So what is the conventional treatment for human lung cancer? First option, surgical excision, in which surgeons remove the cancerous tissue from the body. Other options are radiation therapy and chemotherapy, which have a lot of side effects, such as loss of hair and dysfunction of vital organs. So in this article, researchers investigate the effects of a flavonoid on human lung cancer. This flavonoid is called Cycloartobiloxanthone. It was extracted from the stem bark of Artocarpus comesianus tree, which is a domestic plant of Thailand. And as you can see, it has a cyclic structure and it has a hydroxyl groups in it. In MEDI's part, they used 4 different human lung cancer cell lines and 1 normal cell line to measure the effects of the compound on normal cells beside cancer cells. They designed primary and secondary antibodies for immunoassays, and as I mentioned before, Cycloartobiloxanthone was extracted from Artocarpus comesianus tree. MTT assays was done to measure cell viability. In this assays, MEDI111 cellular oxidoreductase activity is measured. This enzyme converts MTT to formazine. MTT normally emits a yellow line, and it is a cyclic compound, and formazine emits purple light. So these are colorimetric assays. They also made nuclear staining assay to reveal the nuclear condensations, which are big indicators of apoptotic cell death. They also made Western blot to measure the expression of interested proteins. So to measure the cytotoxic effect, MTT assays were done. As you can see, for all 4 cell lines, as the concentration of the compound increased, the cell viability decreased. For example, in this line, it reduced it to 20%. In that, it's almost 0. In this cell line, it is 60%, and this, it is approximately 50%. So we can say that cycloartobiloxanthone has a negative correlation with cell viability, and IC50 values refers to the concentration in which the cell has a 50% cell viability. For example, A549 has 110 microgram. It means that at 110 microgram concentration, it has 50% cell viability. So from there, we can say that this compound induces cell death, but in which way? Is it apoptotic cell death or necrotic cell death? So researchers made an experiment to see this, and it was an immunofluorescent experiment. They used 2 different dyes, hosts and PI dyes. Both are nuclear dyes for nucleic acids, and PI stands for propydium iodide. So as you can see, as the concentration of the cycloartobiloxanthone increased, the nuclear condensation rate increased. You can see from here, and also here compared to normal groups or lower concentration groups. As you can see, there are no nuclear condensation. And also I want to mention that these red dots stand for necrotic cells, and you can see the different combinations of the dyes. So they also measured the apoptotic to necrotic nuclei ratio, and as the concentration of cycloartobiloxanthone increased, apoptotic to necrotic nuclei ratio increased dramatically. So even at high concentration, cycloartobiloxanthone has little necrotic nuclei, and we can neglect that. So almost certainly we can say that Cycloartobiloxanthone induces apoptotic cell death. So, and they also compared the compound with conventional drugs. Cisplatin and Apoptosis was two drugs used as positive control. And in both cases, for cancer cell line and normal cell line, cycloartobiloxanthone has the same effect, almost same effect as the other compounds, other drugs. For example, in normal cell in cancer cells, they have a better effect from Apoptosis. It has 50% cell viability, whereas Apoptosis has 60% cell viability. And in normal cell lines, for example, cycloartobiloxanthone has a better effect from cisplatin, or almost same, I can say. So you can see the nuclear immunofluorescent assays for Apoptosis, cisplatin, and cycloartobiloxanthone, and you can see the nuclear condensation formation for all three lines. Sorry. Sorry for that. And as you can see, apoptotic necrotic nuclei rate was high for cycloartobiloxanthone, and it increased dramatically compared to control group, and it has almost same effect compared to conventional drugs. It has a comparable effect compared to conventional drugs. So from these from the last three experiments, we can say that apoptotic cell death was induced by the compound. But which apoptotic pathway was induced? It was intrinsic pathway, which use mitochondrial proteins, or extrinsic pathway. They measured the concentration, expression of the proteins, apoptotic proteins, intrinsic apoptotic pathway proteins. These are mitochondrial proteins. And as you can see, the concentration of the p53 increased dramatically as the concentration of compound increased. Also, the concentration of the anti-apoptotic proteins, which are MCL1 and BCL2, decreased dramatically, and the concentration of the expression of the BAX, which is pro-apoptotic protein, increased dramatically. So we can say that this compound induces cell death via increasing mitochondria-dependent apoptotic path. Also, you can see from here, cleaved PARP to PARP ratio increased dramatically. PARP normally functions as PARP has a key role in DNA repair, and when it's cleaved, it cannot function. It can't function properly. And cleaved caspase-9 to caspase-9 and cleaved caspase-3 to caspase-3 increased also dramatically compared to control group as the concentration of the compound increased. Cleaved caspase-3 is the active form, and it activates the caspase-3 by cleaving it, and cleaved caspase-3 is a downside regulator of the apoptosis, which initiates the apoptotic path. You can see the Western blot results from here and from here. And beta-actin was the control group. As you can see, BAX was increased, BCL2 was decreased, and CL2 was decreased. And you can see the PARPs and cleaved PARPs. So from here, we can say that pro-apoptotic, anti-apoptotic proteins, pro-apoptotic, anti-apoptotic protein ratio was increased dramatically by the compound. So from there, we also understand that this compound induces intrinsic apoptotic paths. So in discussion part, so the 5-year survival rate of human lung cancer is low. And so we need a safer and efficient therapies compared to standard therapies. So, and the herbal compound is the biggest candidate for it since they are safe. And cycloartobiloxanthone is a herbal compound, and yet it is as efficient as the standard drugs and has similar results as standard drugs. So you can see the possible role of the compound in the inducement of intrinsic apoptotic pathway. It was guessed by the researchers. So they guessed that the compound artobiloxanthone increases the concentration of p53, and it increases BAGs and suppresses the BCL2 and MCL1, and it causes the formation of apoptosis. It forms cleavage caspase-9, and this induces the formation of cleavage caspase-3, and it deactivates PARP, which has a role in DNA repair, and at last apoptosis is induced. So, but what about the toxicity of the cycloartobiloxanthone on normal cells, or the side effects of the compound, other underlying activities on the cancer cell lines? These are the questions that should be answered. So this compound has a lot of effects on the human lung cancer, negative effects, and it is a big candidate for the treatment of the human lung cancer, and it should be focused on further research. Thank you for listening to me.